Okay. We may not spend more than 1 hour, so I want to once again appreciate everyone. I can see Professor Adiola Allah. I can see
Good
Dr.
evening, sir.
Akinola. Yeah. I can see Dr. Akinola Omotayo. I can see Dr.
Amen.
Irene Daniel. Yes. I can see Dr. Mbeneme Azota Odechi. So we are all welcome. Then I can also see myself. So we are going to start quickly because tomorrow some persons might be going to their worship centers. All right. I don't expect much in this presentation. If you notice, a good number of us opted for certificate of participation, where I understand it's a matter of time. So we start with the first on the list, Olawoye Mary. I don't know if she's here. I don't think if she's here. Okay. Then the second on the list is Azota Ogechi. You can share your slide with us. Let's see what you have for us. Azota, are you
Okay.
here? Yeah.
Yes, I'm here.
Okay.
Good evening, sir.
Yeah. Good evening.
Okay. Let's see. Okay. I'm coming. Let me set it up.
No problem.
Okay. Yeah, sorry. Okay, can you see my screen? I don't know if you can see my screen.
Can you rotate your phone so we can see it as landscape?
Yes, sir. Okay. Let me
That
rotate
would
now.
be very perfect.
Okay. Good evening, everyone.
Yeah, good evening. How you doing,
Good
sir?
evening,
Good evening, sir. Welcome, Mr.
Okay.
Azuka.
Thank you. Okay. My presentation tonight is
I'm
on
rooting for you. Right on.
pharmaco. My presentation tonight is on pharmaco-based drug design and in silico working analysis of novel tetrahedral naphthalene derivatives as antitubercular agents against selected proteins of Mycobacterium tuberculosis H378. So this particular Mycobacterium tuberculosis strain has its entire genome already sequenced. So the proteins are already known and the genomic structure has been mapped out by scientists and researchers. So I've used some proteins from this particular bacteria as my uh, test agents basically. So Mycobacterium tuberculosis is basically a bacteria that causes the airborne infection known as tuberculosis. It has a unique structure which makes it quite difficult to identify using simple gram stain and other basic stain methods. It requires more precise and more specific methods like both PCR, acid fast stains, and the likes of it. So as a bacterial infection, basically there are drugs or basic drugs and drugs that could basically stop the spread of this disease, which is known as antibiotics. And these antibiotics have either bacteriostatic or bactericidal effect on the bacteria. And the major issue of the disease is related to the organism itself having certain resistance to different treatments, which are the antibiotics against this particular bacteria. If this case happens, there is what we know as multi-resistant tuberculosis, and even in worst cases, there are the extensively drug-resistant tuberculosis, whereby the tuberculosis bacteria itself is resistant to more than one line of treatment. Because there's a first line of treatment using drugs like isoniazid, rifampicin, ethambutol, and pyrazinamide. If the bacteria becomes resistant to this first line and also the second line, that means there's either total drug resistance or extensive drug resistance. So with the invention or the use of tetrahydronaphthalein, this particular compound has been noted for its multi effects as a drug molecule, because it has both anti-inflammatory, anti-tubercular, anti-cancer, and other various antagonistic abilities or effects against certain diseases known to humans. And also, a fun fact is this particular bacteria is found in almost, or should I say, all human beings, because humans serve as the host for this particular bacteria. The bacteria is basically living in us, but is being suppressed by different, should I say, antigens and various control bodies all over our body, like and the bacteria as well. Other bacteria that live in the human body that help to break down food and also give protection, and other factors like acids and the rest of it. My workflow included one, I had to select the scaffold, which is the tetrahydronaphthalene. I used a prior knowledge on this. I predicted some derivatives earlier for my first project in this Hendeb lab using this particular derivative. So I just utilized the derivative again and decided to design specific molecules from different functional groups to synthesize mine as a form of experimental procedure. And the next step was to pinpoint the modification position on the molecule itself to know where I could attach these new functional groups to stabilize it and use as a pharmacophore design itself. Then the third was to select my functional groups. I had to use the AI permutation scheme, and I had 5 different schemes of 4 positions of functional groups, giving a total of 120 design compounds. Then the next step was to create these compounds by attaching the functional groups to these positions. Then I also had to select reference drugs as a control to know the docking analysis, which I did later on. And the next step was to select the proteins as well from protein data bank, and I had to use a specific selective criteria to screen out a few of the tested or the created new compounds I designed to know if they have some effects on these protein targets, which I will discuss later on, and the molecular docking as well, and some 2D visualization of the ligand protein complex formed at the end. So these are the permutation schemes. In scheme 1, I used phosphate group, ethyl, benzene, acetyl to do my scheme 1 for the arrangement. Then the scheme 2 included ethers, chlorine, hydroxyl, and methyl. Then scheme 3 included aldehyde, iodine, carboxyl group, and phenylbenzyl. Then the fourth one included amide, amine, bromine, and chlorosulfonate. And the last one was nitrous group, fluorine, chlorophenol, and cyano group. So these are some of the design compounds, just a few of them out of the 120. That's the select. Then this was the criteria I talked about where I had to select just 10 out of the 120 for a possible docking analysis. I used the drug likeness feature or lip sync equal of 5 to choose my 10 out of the 120. So the parameters included the molecular weight, the number of hydrogen donors and acceptors, and the topological polar surface area, the log P, which is also the lipophilicity, the biological availability score, the solubility, and the GI absorption. So basically, the molecular weight has to be below 500 grams per mole for easy oral absorption into the body, and the number of hydrogen donors has to be less than 5, then acceptors has to be less than 10. Then the CPSA has to do with permeability, membrane permeability of the drug to pass through various barriers in the body for easy ingestion and also getting to the target point in the body for the effect, for therapeutic effect to occur. And the log P has to do with lipophilicity, which is fat solubility and it has also some hydrophilic reaction to the body. And the BAS, which is the biological availability, has to do with the drug having some kind of biological activity and also determining how the drug will be administered into the body, either as an oral administered drug or intravenous, or depending on the route of administration. The solubility is if the drug is basically soluble in water, and the absorption, the gastrointestinal tract absorption. So these features, there was no violation to any of the Lipinski's rules. The violations were zero. So that's why I said that the codes are written above. I use the minimization energy number that the code names, so I won't know misplace which is which. Then this table included the docking of my proteins with reference drugs. Reference drugs were basically antibiotics known as they are clofazimine, linezolid, levofloxacin, fluoroquinolonic acid, 4-aminosalicylic acid, streptomycin, amikacin, ethambutol, isoniazid, and paraginamide. The docking scores were quite from moderate to high, from negative 6.6 to as high as negative 9.4 for the various antibiotics tested against the target protein. And in comparison, new compounds by design, the scores of the docking analysis of each of them were quite similar, with the new compound 1 particularly having the highest affinity in contrast to the two of them, which is negative 9.6. And that one also had 3 high affinities amongst all the 10 proteins and 10 compounds tested. Then another one had 4. There are 4 highest affinities from a particular one, and the 2 which had 4 and 3 highest affinities are from the same parent compound. Like they have the same similar structure because they have the same functional groups. So the names, I shortened the name here. If you look down, the names are all shortened because the names are quite long. But I had to shorten it. So these 2 compounds with 4 and 3 they were from the same parent structure basically. And these are the 2D images of the drugs. These are the design drugs basically with the various proteins. These are the design drugs with the various proteins. And then these are the new compounds with the proteins. Some of them have similar structures with both the benzene rings and the non-benzene rings attached and other functional groups too. And the interactions are majorly hydrophilic with some hydrogen bonds and also some little we're majorly hydrophobic and also slightly hydrophilic in some of them as well. Then in conclusion, the design analogs actually showed some promise as possible antitubercular agents because they had similar binding affinities with known reference drugs that are used in the treatment of tuberculosis, and some were even higher in some instances. And I noticed that those two I talked about, which were from the same parent structure, they have a particular, from their nomenclature, they have a particular kind of acetic acid, which is known as phenoxyacetic acid, which makes a compound have some effect against the bacteria itself and can possibly kill the bacteria if this compound is present. So the acetic acid combination with a phenyl and oxoethyl group makes it more potent in possibly eliminating Mycobacterium tuberculosis, which serves as an evidence to why they had the most affinity to the various protein targets from this bacteria. And in recommendation, I would say that further studies could be employed to maybe determine the predictive and interactive model via like molecular dynamic simulation to know how toxic and how compatible these new compounds could be in both in vivo and in vitro interactions, to with also clinical testing models to know how stable they are and how effective they are without causing either genotoxicity, cytotoxicity, or any kind of toxicity to the host. And also possible synthesis of these compounds, because if these compounds could be actually synthesized, then they could actually be tested in vitro and even in vivo and know the effects they have. And yes, thank you for listening. I hope I didn't bore you people.
No, you didn't.
You did not.
Okay,
You,
thank you.
Thank you.
in fact, what you did now, I don't know, but others should not follow you because you disobeyed. You just disobeyed the like what will I say? You went extra mile, and you know that, and I appreciate you for that.
Thank you, sir.
Because we are just expecting that you model these compounds and show us what you have modeled. But I appreciate that you now took the compound you have modeled for docking, and I wish, that was not part of the instruction, but you did that
Mm,
on your own. So I will appreciate you for that. And
thank you.
I was just thinking, have you done docking before, or you did that from all this our small
Yes,
training?
I have.
Okay,
Yes,
you do.
yes.
All right. But I was shocked if it was this small 2 days training that we were able to do that. Okay. Uh, did you have any challenge while doing the work?
Well, initially, when I was, trying to develop the functional group, some of them were not present in the chem sketch, so I had to look for alternatives. And then sometimes when I include it, when I'm trying to generate the title of the compound, it might tell me little errors here and there, but I eventually overcame those errors. So
Okay.
those are the only
And
challenges.
yeah, I noticed you represented the identity of the compound with their minimized energies.
Yes.
Okay. But when you do the compound in time stretch, were they able to was the time stretch able to generate their names?
Yes, and their CIDs, their smile codes.
Okay. And you save them with the name, right?
Yes. Although some of them were like inverse of each other. Like
Okay.
maybe the numbering of the positions were literally the same for some compounds. So I used inverse to rename some of them. That's this
Okay.
3D structure.
The point I want to clear is that some persons must have also encountered the challenge. When you draw a
Mm,
compound, let's say I draw benzene now in time stretch,
mm,
then I take it to 3D and save it as a mol file. Then, so
Yes.
if I save it as mol file, I actually saved it with the name. I saved it with base. But you see that while you are doing your docking, those compounds did not. I'm not sure if they opened. Did you use Pyres for docking?
Yes, I did.
So when you open them in Pyres, were they showing the name you used to save them?
No.
So how are you able now to identify that this one is for this one?
I had to write it down. Make sure I checked everything and I wrote everything down. And
Okay, that's actually a big challenge. Yeah, yes. Why I'm saying this is because I noticed you now use their minimize energy. So there's a way out. There's a way out in which you can save a compound with the name, and when you open it in Pyres, it will show that same name.
Okay.
Yeah, there is a way out. In one of the trainings, because I shall be explaining that aspect in green, yes, in green chemistry, yes. I shall be explaining that aspect in green chemistry. So, and also in the training coming forth next week on computational chemistry for beginners. So we shall also look into that aspect. Yeah. So that is what I wanted to point out because I noticed that you presented with their binding energy. And then I have a question. The question is, what was your choice? Like, what was the reason for your different scheme? Did you just develop those schemes like the groups you use? Did you just use them by let me not test nitro group, sulfonyl group, and so on? There was a reason, there was maybe a scientific reason that made you choose those functional groups you use in your scheme.
Okay. So first of all, when from the whole class, you made mention of various features, both the hydrophobic and hydrogen bond and the rest for pharmacophoric features. So I had to start searching for when I was looking for the structure of the parent molecule, which is the tetrahydronaphthalene. I had to start looking for possible functional groups that could bind to this particular structure and cause some kind of effect depending on the effect you're looking for. So I saw different examples. Like I said before, I saw there are so many functional groups I wrote out before, but I couldn't find them on the ChemDraw. So I have to substitute for either closer relatives or something completely different if I don't find a particular close relative to this kind of functional groups. And then I noticed the halogens work well with this structure. Then also the amide and amine, because this particular amides and amines give it more effect depending on the nature of the effect you're looking for. So these are basically my reasons.
All right. So lastly, in your docking, did you also dock with the parent chain, or you docked only with the optimized ligand? What I mean is that you use a tetrahydro what?
Naphthalene.
Yeah, tetrahydronaphthalene. So did you also dock with the tetrahydronaphthalene, or you left it and only docked with the ones you have optimized?
I docked with the ones I've optimized.
Okay. The reason is so that I wanted to know if the optimization was better than the initial compound, so that we can know, okay, see the initial compound tetrahydrophenol, see the binding affinities, and compared to the ones we optimize, we can now tell that the optimized compounds we are better than the initial parent compound that we optimized. So I would have loved to see it. Yes. Do you understand my point?
Yes. Although before I worked on this when I was predicting different derivatives, I used different derivatives of the original tetrahydronaphthalene and did some binding affinity on it. So I'm trying to look for the this thing if I can find the result of the docking I did that time. So
Okay.
look, yeah, I
Well,
did something like that before.
all right.
Um,
Nevertheless, you have done well. I really
Thank you, sir.
appreciate your effort. Yes. So we can tell that the optimized compounds were likely better than the original compound, and that is
Yes,
actually our aim. That is our aim. Thank you so much. All right.
thank you, sir.
So if I should follow the list, we have Daniel, Irene. These two names, Daniel and Irene, it to me, I would say, is he a male or a female? So I will now know. I'll be wondering. All right. Then Irene, are you here with us? I saw that name as part of the people who were present. Daniel, are you with us? Yeah, he's here. I can see the name.
Yes, I'm with you, sir.
Okay, Namano.
No, man.
Omo. I, you know, so many names are shocking me. Oh. I I I always I thought that Irene was only for females.
Uh, yeah, you're right. That's my native name, anyways. So it's not
Okay,
pronounced.
okay, because in Igbo I used to call it Irene.
But yeah, that's the correct pronunciation, actually.
Eh, but people
Irene,
don't agree that it is Irene. Oh.
it is.
They say it's Irene. Nevertheless, you can go ahead to share your screen.
So I have a little bit of challenge right now.
Okay,
I'm using my phone, but the document is not on my phone.
okay,
The
okay.
PowerPoint.
You did docking.
No. I didn't do docking. Just follow the guidelines you gave.
Okay, so do you have the PowerPoint in your phone?
No, I don't.
Okay. Okay, but can you share with us can you just tell us what you did?
Okay, that would be fine. Uh,
Just
okay. I I worked on indoor scaffold. During the training, the illustration you gave was benzimidazole. So I went online and I was checking, and I saw indoor scaffold, and I was like, this could, though they kind of have similar structure, but they are different. The indoor and the benzimidazole, they are similar but quite different. So I worked on the indoor scaffold, and indoor alkaloids are a class of alkaloids containing a structural moiety of indo, and plant-based indo alkaloids are very rich in pharmacological activities, and the endonucleus is considered to contribute greatly to these activities. Um, Many of the therapeutically active indo alkaloids are isolated from plants, and these compounds have had a noticeable impact in medicine. Right. So I used 5 schemes, scheme 1 to 5, with different functional groups. I used the main functional groups, the CH3, the chlorides, the carboxylic acid, the ether, and the likes. So they are in my slides when you go through it. So this the scheme I used in um, in getting the 100 analogs. Asked us to get. I don't really have much to say. All of the schemes and the analogs are in my slides. It's quite unfortunate I can't share them now. Okay, what else? What else? What else?
Can I
You said
get your slide? You submitted your slide, right?
I sent it to your yeah, I sent to your mail. Yes, sir.
Okay. Okay. Let me see how I can get your slide
Okay, sir.
because I'm interested in what you are saying, so I need to see your slide.
Okay, sir. I sent to your mail, sir.
Yes. So what I will do for you now is I'm going to open up the slide. So you will come back. You will come back to just explain the slide. So why I call on Ines Adiola Allah, Professor Adiola. Yeah, Prof, are you there?
I'm here. Can you hear me, sir?
Okay, yes, I can hear
you see
you.
my screen? Can you see my screen now? Can you see my
Okay,
screen?
yes. Yes,
Okay.
yes. Yeah, present.
Okay, thank you for having me. I'll be speaking on pharmacophore-based design of 2-heptanol benzoate derivatives from the workshop, the mini presentation from the workshop that we just finished. So I'll be going according to this outline that I wrote here. From the workshop, we were basically being taught about drug design, structure-based drug design, and ligand-based drug design. The first one uses the 3D. Sir?
Prof. So for you to see your slide very well, just help me
make it
click on slideshow.
Yeah.
So
Slideshow, yes.
Thank you.
Sir. Can you see it better now?
Perfect.
Okay, so I'll be speaking to this outline. From the workshop, we are basically taught about drug. It's all about drug design. It could be structure-based and it could be ligand-based. So this particular one explores just ligand-based drug design, and the difference is that in SBDD, the 3D structure of the target, like a protein, is used to design the drugs. But in what we are presenting now, we use ligands, compounds that are already known to bind to a target, to design new drugs by just changing the functional groups, like we've been hearing. So I won't talk about the SBDD. I just defined it here. But the analogs in the ligand-based one, if you have a known ligand, if you have a known active ligand, like my own original compound, I can modify it by changing the functional groups R1 to R4 positions to improve activity. I already did docking at some point, so I just used one of my analogs, but I'll explain. So the analogs could be part of either approach, depending on whether you are using target structure or modifying for non-ligands. Like I said, this particular work is about the ligand-based drug design. So, in the docking that I did, I got a, okay, in the docking that I did, I was targeting urinary tract infection, and I was using clove. I had clove ligands. So I already got some hits, like maybe four or five hits, and that looked likely. So in this work, my active ligand, my original compound from a former docking, I modified it, changing R1 to R4 positions to improve the activity. So in this ligand-based one, pharmacophore is a set of features like the steric, electrostatic, H-bonding, essential for bioactivity. Pharmacophore set of features, they're not really for ADMET, like toxicity. I hope I'm right. That's what our teacher taught us. I'm just trying to remember. Then the key elements of pharmacophore features They are the H bond donors, the acceptors, the hydrophobic, the hydrophilic groups. So the aim of this study is pharmacophore-based design of 2-heptanol benzoate analogs to optimize binding to protein, 4CA4, PDB, all that, structure of fimbrial H-lectin. Please let me just quickly explain. E. coli, in the origin, E. coli causes urinary tract infections in human beings. So I tried to use clove, the plant that is called cloves. I got some ligands from clove to try to attack the E. coli so that it will not be able to bind to the urinary tract of the human being, and therefore there won't be any infection. So that's what it's all about. So the E. coli adhesin found in pathogenic E. coli, the bacteria, is involved in the adhesion. So that's what the target was when I did the docking, to be able to prevent E. coli from attaching so that there'll be no infection. So briefly, the 2-heptanol benzoate was a ligand that I found to be good. Is the heat scaffold reference drug from my past docking study, not studies one, from my past docking study. So the first thing I did was to identify active compounds. In this case, one. I tried one first to gather the ligand. Then I generated pharmacophore models, identifying the common features of the pharmacophore needed for activity. Then I tried to design new molecules using the model we were taught, with those groups, with those side chains, to see whether I can get new compounds. And the last part of it is that I should test the new compounds for activity in silico, like docking, in vitro or in vivo, like in the lab. But this will be further work because all that will not be possible. So I identified my reference drug, which I have mentioned. Then I identified the positions to be modified. Then I identified the permutations for each scheme in a tabular form, which we'll see later. Then I drew the analogs and I generated their names and smile codes like we were taught. And then I converted the structures to 3D for the presentation. So I used like I said, I already had a heat ligand from the previous work. So I had these 2-heptanol benzoate, and that's the structure. So I now decided to optimize the ligand. So I have my R1, R2, R3, and R4 positions. I had those ones. Then I first of all did manual arrangement of those side chains. I think it's here. But then our teacher said we should use AI. So when I used AI, I discovered that I first got 2 possible analogs with different positions of side chains, R1 to R4 positions. I first got these ones. Then I asked AI again, that could I get more? It gave me 24 more. So I first got 24, then got 24 more. Then I asked AI that, what's the maximum amount of analogs I can get from this R1 to R4 positions? So it told me that it's a permutation thing, like our teacher said, and that I can get up to 256, possible different positions. So that 256 was very, very scary. I just started with what I had. So like I said, for my workflow, using ChemSketch, you use the tools, and then you use the tools to get the smile codes and the name. This is analog 2 because analog 1 was my original 2-heptanol benzoate that I had before. So analog 2 is after I had changed a side chain. So I now got this is the 3 structure that I got. But I discovered that the scaffold is disconnected. If you see, there are 2 red ones on top and the ones at the bottom. And I was not wondering that what kind of human being without neck, because I can see 2 heads and body. I don't know what's happening in the middle. Then so when I now tried to generate the name, it was telling me that the name is not on PubChem or something, but it gave me the smile code. So when they gave me the smile code, I had serious problems with this 2-heptanol benzoate. One problem after the other, after the other. So I now discovered that maybe the problem I'm having is because of the structure, because it's really disconnected. I tried to check my structure, the scaffold. I discovered that in the work, it's the 2D structure is connected, but when I do 3D, it's not connected anymore. So in my frustration, what I did was to change it. Now, I told you I was working on urinary tract infection. So that, my heat compound that I used was what I got from the ligands that I docked with. But the urinary tract infection, they normally treat it with ciprofloxacin. So PopChem, I did all that, and I got this 3D structure of the 3D structure, and it gave me not just the structure, it gave me the smile code, everything, because I'm working with a known drug. So I decided to just change everything to this known drug. So this is because I now changed from my original heat compound to this known drug, which I also used in the docking. I think it came first, it was the best. So I got the name, I got the smile code, and then I had all those R1, R2, R3, R4 side chains that I was going to be trying. So I worked with the first analog, and I got the smile code. Then the software told me that it cannot generate a name for me. That once a name is more than a certain number of letters, it's impossible. So I had to run with this. And since I already generated from AI all those side chains, I decided to start trying them one by one. But quite unfortunately, my laptop has been hanging and hanging and hanging. It will freeze for long. I've been struggling with it, and I was able to do those. I was able to get analogs, which I put. I got some analogs which I put already in a folder, but I wasn't able to do as much as I thought I would be able to do. So I couldn't generate too many. All right, I have about 10 analogs or so, but not really many, because I've been fighting with this laptop and I haven't been able to fix it. So I want to thank Mr. Harry so much, because honestly, I think this is a quantum leap from where I was before when it comes to knowledge, computational knowledge, you know. So I really want to thank him and Hendeb Industries. And now, if you don't mind, I would like to share also let me try and see whether I can share my folder that has those the analogs, whether they are dead or alive. Let me just, yeah, okay. So I have this is my folder. So I had the challenges that I had was that one major challenge was when my structure refused to conform. The first ligand that I used, when it refused to conform to what we were taught in class, and I had to change it. Please, can you see my screen?
Yes,
so I have,
yeah, we can
so
see.
that's my folder with the few analogs I was able to come up with. Thank you very much for listening.
Such a wonderful job. It was funny that you had a structure without head.
Yeah, I don't know. I can't understand. So I had to change it.
Wow. The major concern is that you have the principle behind what you are doing, and despite the challenge from your system, from everything, you still keep it up. So because you know what you are doing, and that is the most important thing. And I know that if all things being equal, you are going to finish this analog to 100 by God's grace on your free time.
Amen.
I trust you. I know what you can do. Yes, I know what you can do.
Amen. I hope to continue.
Yes, I really appreciate that. Yeah. So, like I've said, to be perfect in the chemical structure representation, the next week training will be very, very helpful to everyone, because we shall take our time to teach how you can draw those things you used to download from PubChem. It was deposited by somebody. So we shall learn how they do it like that, and how they model structure to be perfect to that standard we download from PubChem. Yes, so we shall, and we'll be using ChemDraw for that, because ChemSketch has limitations. So once again, I appreciate our prof, Professor Adeola. She has been a wonderful friend to me, and well, I will reserve my comments.
Thank you, sir. Thank you so much, sir.
Yeah, thank you so much. Thank you. So I will share the screen for Daniel. Daniel, can you come in?
Yes, sir.
Daniel, are you still?
I'm with you, sir.
Okay, I will share your screen, so you will just be fast with it. Can you see it?
No. Oh, yeah, I can see it.
Okay, I can see it. All right. So you be telling me next why I do it for you.
All right. Like I said earlier, I worked on with the Indo scaffold, trying as much as possible to get several derivatives. Yeah. All right. Like earlier said, Indo alkaloid, a class of alkaloids containing a structural moiety of Indo.
Yeah, I think
And
you have cleared this part now. You have done this
okay,
part.
just next. Okay, just
Yes.
uh, so now we're represented. All right, this just my schemes. The schemes are used in getting those analogs, the 100 analogs, scheme 1, scheme 2, scheme 4. Next slide, please. Scheme 5. Yeah. So I worked with a main functional group, carboxylic acid, ether, ester, hydroxylic functional group, HSA. Next slide, please. Okay, this the table containing the first scheme analogs. Like you taught us, I used AI in generating these 24 analogs. Right. So I worked with 4 arrow the arrow label, yeah, arrow 1, arrow 2, arrow 3, arrow 4, which I'll see later. I kept some structures down the slide. So I was just basically rotating, these functional groups around those labels. After which I generated their names and their SMI codes. Then afterwards, I converted them to a 3D structure. Okay, this for okay, fourth scheme. Next slide, please. Fifth scheme. Okay, these are some of the images of the. Okay, this first is analog of scheme 1. So the arrow 1 is this methyl group there. The arrow 2 is this ether group there, OCH3. Okay, arrow 1 is the main functional group. Arrow 2 is the methyl group there. Then the ether group is the arrow 3. Then the chloride is the arrow 4. So I was just basically rotating the analogs AI gives to me. Next slide, please. Okay, this for um, scheme 2. The first is analog of scheme 2. Next, please. All right. So pharmacophore-based drug design is a helpful approach to help design compounds that could be a potential drug candidates. Pharmacophore-based drug design also helps with lead optimization, thereby reducing the aspects of these compounds that are responsible for toxicity. The Indo scaffold is a good starting point to derive compound that exhibits anti-malaria, anti-inflammatory, anti-cancer activities, and the likes. So pharmacophore-based drug design, when combined with AI and machine learning, could revolutionize this aspect of drug design. Thank you, sir.
All right. That was a very wonderful one. And it gives me joy when I see these things. So, but what was the choice for your stream? What guided you in choosing your stream?
I looked at this indomethacin. It's an indo derivative. So, and indomethacin is for long now because of the GI effect it causes, the GI bleeding. It's an NSAID actually, anti non-stress inflammatory agent. So I just picked it with the hope that if we could just rotate these positions, maybe we could have a better drug for it. That's what influenced me, sir.
All right. Thank you. You did a very good job, and the pictures of the images shown that they were well drawn, and that is a very good job. So subsequently, further studies, you can now deploy them in your doctoring studies to see their activities. So once again, I appreciate that work.
Thank you, sir.
Right, I will call him Amoke Victor Chukwemeka.
Okay, good evening, sir. Thank you very much, sir. Can you hear me, sir?
Clearly, I can.
Okay, I think let me share my screen.
Okay. Why Amoke is doing, let me be sure. Go ahead, though. Just go ahead. Akinola, okay, yes. Akinola is present with us. Jeremiah, you just sent your work. I don't know. Is Jeremiah here with us? Jeremiah John.
Hello, sir.
Okay.
Please, can you see my screen?
Hello, sorry,
Yes.
please. I want to say something, sir.
Okay, go ahead.
Yes, sir. Please, my name is Omatayo Akinola. So sorry, just the way you do for the other guys, sir. Please, I'm having an issue with my laptop, so and I'm currently on my phone now. So probably you will also help me to show it from my own side, from your own side, sir.
Okay. Thank God you requested that very early. So
Thank you, sir.
I will do that now. All right. So
Yeah,
Victor, you can go ahead.
Can you see my screen, sir?
Yes, I can see the screen, though
I'm using
it's
phone.
half.
No, that's the problem.
Uh,
Very serious. Even though I have to put it in landscape.
yes, it's good that way.
It's supposed to be full.
It's supposed to be full. That is what I'm now looking at.
Did you try zooming it out?
I zoom it
Did you
out.
try
Is it full now?
Make it upright. Let's see. Make it upright.
Let me put it in last.
No.
Okay, let me do that. Let me make it upright.
It's like we see it better like that.
Okay. I don't know.
It's upright, not better. At least it's showing more. We're seeing more of it.
Yeah.
So it's small.
Okay, no problem. Let me. Okay. It went back to. Okay. Sorry.
It was good now. This is okay.
Okay.
This is okay.
So what I did was I had to do analogs of metformin. My work is mostly on anti-diabetic research. So metformin is one of the first line treatment in diabetes. So I have to work on it. So metformin is an anti-diabetic. Sorry, metformin is an anti-diabetic. Can you hear me, sir?
We can hear you.
Yes, we can.
Sorry, call wanted to distract me. So metformin is an anti-diabetic agent used for treatment of type 2 diabetes. It's a first-line prescription for management of type 2 diabetes. Metformin belongs to a class of drug called biguanide. It helps to control the amount of blood sugar in the blood, as glucose in the blood. It decreases the amount of glucose in the body absorption from food and amount of sugar made by liver. It also increases body response to insulin. So the most important thing, for me, most of the times, due to some challenges, so is it needs some like sulfuria to combine with it to make it more functional or more effective. So that's why sometimes they combine it with glibenclamide. So what it informed me to find a way to develop an analog that can do better without combination, so that it can go single dose without increasing the dose. So what informed me was to develop an analog that's without high dose, it can also do the function it's supposed to do with combination. So that was what informed me. So due to the side effects that can arise as a result of use of metformin, like gastrointestinal issue, reduced appetite, vitamin B2 deficiency, lactic acidosis. So developing an analog can reduce side effects, which can be very helpful. So the development of analogs that has more potency can help to reduce they can use the drug without combination, without combination with anti-diabetic drugs, which can cause more side effects. So I developed 100 analogs. Sorry for the mistake for analog. I designed 100 3D optimization using ChemDraw. The structure. So some of the structure, due to the long name, so I couldn't generate their name, so I had to use Smicle to generate them. So I used the scheme, these 4 schemes, even though the 4 of them using AI gave me a total of 96. So to complete the 100, so I have to interchange some of them to get the 100. Like for example, I use scheme 1, I remove one molecule. I mean one functional group from one, maybe R1, combine it maybe with R3 in D to get what I need. So that is how I arrive at 100. So this is the structure. The left side is the structure of the metformin, and this how did they call it? Sorry, I don't know why this thing is not clear. The general structure of the analog. So where I put my analog, my analog is R1. I don't know why the thing is not clear here. So these are the structures. Some of the structures are generated. I have hundreds of them with their names, and I didn't need to explain most of them, but this is the 3D after optimization. This is the 3D. So, and what I found out, so after the whole optimization, so I had to do analysis to confirm the toxic effects. So I have to do physiochemical analysis and toxicology analysis to using ADMED. So even though I didn't present it in my data because it was what I had to do at the end of the work, because I wanted to confirm what happened. So I found something interesting. The reason why metformin is the only drug mostly used as a class of biguanide in treatment of diabetes, type 2 diabetes. Like phenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenylphenyl analog of metformin because of the toxic effect. So when I did my analysis, I found out that most of the analogs I created have high toxic, when I have to do this, have high toxic effects. So it's only 3 out of the 100. I think it's only 4 out of the 100 that is good. I have to go to PubChem to download analog, also analog of metformin also, and the same issue. So I had 106 analogs from PubChem, the same issue. When I analyzed them, I found out that they are toxic. So that now is for me the reason why most they cancelled production of some of analog of metformin. They removed them from the market. So the analogs has somehow toxic effects. So that's what I found out. And based on the study, only analog was designed and modeled, which can further to be analyzed to get a heat analog with better pharmacological properties, especially the pharmacokinetics property. This can help to develop analog of metformin with better activity and less advanced effects. So in that regard, sir, I want to solicit if you can give me idea how to, it says something about is it biosulfur? I can't remember the word you use anyway. Changing a particular drug that has toxic effects to make it more potent. So maybe something that I can work concerning this metformin, because when I did the work, it was interesting study, and I need to further on it to know how to make it more toxic. Thank you very much.
All right. You have done so well. You have done so well, and I really appreciate that. I was interested in the novelty of the work, and you just explained the novelty of the work, and that is great. Now, I like the way you did your screening. So I like the screening you did, the toxicity screening. That is very important. That after we have done design, we have to now subject all those analogs to ADMET test screening to see the ones that we're able to pass and the ones that are better than our original compound. So you did a very good job. All those parts will be incorporated in our March training, because by March, we are putting up a very big training, the whole of March, that will cover some of the things we did in Pharmacophore and more, the things we discussed in our webinar, and the things we did in modeling. So we are putting everything for March. So you asked a question about the bioisoster, because I explained from the work we did on aspirin, we noticed that the carboxylic acid group of aspirin was responsible for a particular toxicity. So we have to develop functional groups that, number one, we replace that carboxylic acid group and we not alter the activity. So it's a very critical job. We want to maintain the same activity of the aspirin, but we don't want to make use of that carboxylic acid functional group. So we'll be down to screen as much as possible some of this functional group that will replace the carboxylic acid group and still maintain the exact activity of aspirin. So we are including that training also for March. So I will encourage you to just look for 20,000 instead of 50,000. Yeah, thank God Professor Adiola is there. So she knows what people pay for monthly training like this. She can really testify what people pay for monthly training.
Yes.
But we are subsidizing all our monthly training for just 20,000. So I won't like to say anything, but some of us know.
Thank you.
At least most of us know what they pay for monthly training.
Yes.
So yeah, so we are just subsidizing it for 20,000, and this thing comes up every month. So I will appreciate we killing for that March training. There we are introducing this bioiso step. So thank you very much for that work and the challenge you encountered. I really appreciate. Okay, Akinola,
Yes, sir.
I'm going to share your
Yes, sir.
screen right away.
Yeah, thank you, sir.
Jeremiah, Jeremiah, are you here? If you're not, so that I'll just know that after Akinola, I will prepare to visit my bed. Jeremiah, are you here? Okay. So Akinola, you are the last presenter. Glory be to God. So that I really need to sleep. Do I will sleep this weekend? This Sunday. I've had a hectic day, seriously. Okay, your screen is up.
So, yes,
Can you see my
I can see it, sir. Good evening, everyone. Yeah, Dr. Henry, thanks for this golden opportunity. I think with time we are learning, sir. So, my name is Omotayo Akinola, and I'll be talking about pharmacophore-based design of chloroquine derivative. It's a mini project. Please, let's go to the introduction. So, malaria, because actually working on, I worked on chloroquine because of the fact that during those days it was used in curing malaria. You know, malaria is one of the major problem concern in Africa. So, and that's why I chose the chloroquine. Pharmacophore-based design enables systematic modification of lead compounds. So I selected functional groups that were strategically introduced into chloroquine to generate novel derivative. So, and just like our instructor said, he said I use artificial intelligence tool in order to predict a possible substitution region of the chloroquine scaffold. So, and I discovered that I use 5 different groups, and by the time we calculated it, as in 5 factorial, just like what our instructor said, I got 120 different ways of doing that. So, and out of that 120 potential areas, 103 were chemically reasonable position, which were selected. So, out of that 5 groups were the hydroxy group. I chose the carboxyl, I chose the acetyl, I chose the amino acid and the ethyl. And sorry for the way I grouped it. So those 103 potential areas were what I actually showed on my this thing. So as you can see from there, I then try to. Hello, are you all there, sir?
yes.
Yes,
Hello.
sir. We can hear you.
Okay,
Can hear you.
so I try to show the structure of all the possible molecules that were generated. So the dimensional, because of the fact that it is important in the pharmacophoric feature relevant to the biological interaction. So those are my structures. Most of part of the structures that I try to generate out of that 120, I showed part of this. And let me just go to the conclusion. Pharmacophore-based design was actually successful when I used chloroquine using assisted AI in order to generate those viable derivatives, and I modeled them in 3-dimensional way. So though I am just trying to learn this part of the course, so I'm not that good with docking, which my other colleagues have already gone ahead, and the had made prediction, and even the biological evaluation will be carried out to identify most of the promising candidates. Thank you so much, sir.
Yeah, welcome.
Yes, sir.
I would like
Yes, sir.
some of us to comment on his structure. Just look at this one here, as somebody should comment on it.
I use the scheme sketch so to try and draw the structure. So and I use it to generate the
Yeah.
smile.
Your structure looks like me.
Ah.
Oh, it's extending towards the heaven.
Okay.
I can see the non-uniformity or in bond length, but nevertheless, you have done well.
Thank you, sir.
Yeah, you have really done. It
Thank you,
shows you are learning. Yes, it shows you are
Maybe
learning.
Ah, thank you.
maybe that is why my own structure didn't have neck.
Voila.
Hey.
But we're learning.
Yes, we are learning. No, we are learning. We are learning seriously. I want to appreciate everybody. Thank God we are able to make use of the time. Yes,
Thank you, sir.
of time and we're able to make use of.
Hello.
Yes. I want to
Hello, sir.
thank you once again. Yes.
Hello, sir. Am I permitted to ask some questions, sir?
Yes.
Do we have any? All right. Yeah, I can hear you. Yes.
Yes, sir. I'm sorry, I'm not able to make
Go ahead.
presentation, just like I explained to you on WhatsApp, sir. But I want to ask some questions.
Yes.
Part of my questions is about saving the analogs on the Chem Sketch. I got some analogs, but I wasn't able to save them on Chem Sketch. I was just copying them and saving them on a separate Microsoft Word page.
Saving the structure. What?
Because part of what you asked us to do is to save them as ACD format. Apart from the MOV file, you said we should also save it as ACD.
Yes. The ACD now is the whole everything you have drawn in that claim sketch. You save it as the claim sketch file.
Okay.
You first of all draw everything now in the claim sketch before you pick them one by one to save as more file. So that entire structures you have drawn in the claim sketch too. We expected that you save the file also.
Okay.
Yeah, that is it.
Okay, sir.
But the most important one is the individual files, the individual structures you save as more file in 3D. That is very important.
I did that.
Yes.
Another question, that there are some molecules that the Chem Sketch cannot get me their names, but was able to get the smile notations.
Yes, it's possible because Chem Sketch cannot give name for large number of atoms. So mostly 100 number of atoms. It will not generate
Yes, sir. I had that problem.
Yes. But Chem Draw, which we're using for the foundation training next week, Wednesday. Chem Draw can do all that. In fact, Chem Draw can generate spectral data like NMR data. Chem Draw can do a lot. So Chem Draw can generate any name you want, and that is what we'll be using.
Okay, sir.
Chem Draw training.
Okay, sir. All right, sir.
Yeah. Do you have another question?
For those that didn't register for the for that something before it closes, can they still register?
Yes, because we want to send out mails tomorrow, being Sunday, because the training is starting Wednesday. So we want to send out mail tomorrow so that they can use tomorrow evening, Monday and Tuesday to download their softwares. So you can still register.
Okay, sir. All right, sir. Thank you, sir.
Welcome.
Yes, sir.
Hello, sir. Can you hear me, sir?
Yeah, I can hear you.
Please, my question is, when I finished everything, when I saved my own listen to SDF, that is structural data file.
Data file. Yeah.
It was difficult for me to dog. I asked a question about, after presentation, you said that ChemScale doesn't allow minimization of energy, if I can remember. So
Yes.
we're trying to minimize the energy and everything, just normal docking. So after everything, it was showing me error. Even though the name, just like what you commented about the name was not shown. Like the name, like after this, due to it doesn't have CID code now, so it's only showing the chemical name, maybe C2H04, something like that. So I don't know what to do to do that.
Yes. There is I commented on that before from the first presenter. There's a little tactics, a little pattern we'll do to now make sure that anywhere the structure appears, the name we saved will appear. Yes. Otherwise, if the structure appears anywhere, even if you saved it with the name in your normal folder, but if that structures, if you upload it in Pirates or Discovery Studio, it will not show that name you used to save it. So there's a little tactics, a little just little thing you will do to it to make sure that it will now show, so that you can easily identify it if you are dotting that this is this is this and this is that. And that is what they do in PubChem. Now, if you download any structure in PubChem, it carries the CID. And even when the name will show conformer, a 3D conformer CID number. But if you now open it in Pirates, it will not be that conformer again. You only see the code, only the code. That means that they did something to that structure that will make it anywhere you open it, you just see the code 442 or 1127. But when you downloaded it, it was not only bearing 442, it bears the 3D conformer CID 442. But when you now open it in your docking software, it will only show the number 442. So these are part of the things we shall open you up to, we shall learn by next week. Also, it's part of the 3D modeling. So it's part of what we'll do next week. And it's also part of the training by March. It's still part of the training by March. And then so yes, that is the question. So then you asked about the minimization. Now, even if you were able to minimize with ChemSketch, if you're using Pirates for docking, Pirates also does minimization. Yes, Pirates also does minimization, and minimization is also part of what we'll do next week. I think that is the last. If you look at the flyer, there is minimization and optimization as part of what we are doing next week in the beginners training. So you can try your best and get that training. So that training we cover up everything you need to know about structural modeling, about structural drawing and representation. That is the essence of that beginners course. So I think it will help us a bit. To help us a bit. By next week or by tomorrow, we shall be releasing our February training, because we shall be releasing the training we have for first week of February. I think, let me just say it here. First week of February. Let me see what we have for that February. So if I can't show you, that means I will just the
green chemistry, sir.
Flyer will be out. The flyers will be out by February first week. So let's also get, yes, yeah, green chemistry is for the 1 month, the whole of February. The same way we had workshops. The same way we had workshops, webinars, two free training workshops and webinars. So that's also how we have for the month of February. I wanted to lay my hands on that. Okay. So just get prepared for us because we have a lot to offer. We have a lot to offer for February. The same way you joined this January training, don't also get relaxed. I think first week of February, the first training is QSAR. That is the training for first week of February. I wanted to just open our calendar and show you, but it's taking me time. So that's by the way. So at this point in time, I want to appreciate you all for coming around, and I want to say thank you. So you'll be seeing our flyers back to back for February. And also I would encourage you, if you have 20,000, join us next month, next week, or next 2 weeks for green chemistry. It's novel. It's still a novel approach, which I can say no, you can barely find work on in silico studies in green chemistry online. Currently now, you barely get a work on it. So if you can join in that 1 month training and get yourself acquainted, you can start making publications. Maybe your paper might be among the first line papers on the green chemistry using in silico tools. That is coming by 10th of February. So once again, thank you all for being here. The certificate will be made available from tomorrow. Yes, from tomorrow. So we had just 7 persons who prepared their paper, and more than 30 persons maybe did not have the chance as well. So once again, thank you. Thank you. So
Thank you, sir.
we shall meet again for training first week of February. Plan will be out. It's a free training first week of February. We'll get a flyer. We appreciate you for always sharing our flyers, our advert. We say thank you also for that. So at this point, good night. Good night. Good night.
Good night, sir. Good night, everybody.
All right.
Good night to everybody.
Good night, sir. Dr. Henry, good night. Good night, sir.
Yeah, good night.
Good night, doctor, and good night to everybody.
Good night.